Perfect. Thank you, Jara. Um, Thank you, everyone, for joining us today for the Institute of Bioinformatics seminar series here at UGA. Today, I have the pleasure of introducing Dr. Mandev Gill. Mandev Gill is an assistant professor in the Department of Statistics at the University of Georgia. Before arriving at UGA, he completed his PhD at the University of California, Los Angeles, and then worked as a postdoctoral researcher at KU Leuven in Belgium. His research focuses on Bayesian statistical and computational methods for studying infectious disease dynamics. Please join me in welcoming Dr. Gill, and I look forward to your talk. Thank you.
Okay, thank you. It's really great to have this opportunity. So let's start with the idea of a phylogenetic tree. So as most of you probably know, a phylogenetic tree is a directed graph which represents the evolutionary relationship of related species or populations or individuals or genes. And for our purposes, we're going to be primarily interested in inferring the relationship between pathogen sequences. So we can use an evolutionary model to estimate a phylogenetic tree from molecular sequence data. And we think of the data as corresponding to the tips of the unknown tree. And we can think of the internal nodes of the tree as corresponding to common unobserved ancestral sequences. And we'll get into more detail about this later. So this kind of phylogenetic reconstruction forms the core of what's called or what's come to be known as phylodynamic inference, which aims to understand the interaction of evolutionary, epidemiological, ecological, and immunological processes. And this is possible for rapidly evolving pathogens because their evolutionary and epidemiological dynamics occur on approximately the same timescale. Okay, so a phylogenetic inference method is especially useful because it can reconstruct certain events and processes that shape epidemics and outbreaks that are otherwise unobserved and that you can't quite obtain using other methods. And there are a lot of important questions which phylodynamics can help us answer. So, for example, how is a pathogen evolving and spreading? How far has it spread? When did it spread to different areas? How big is the outbreak? What are its circulation patterns? What are the factors that are related to its evolution and spread? How have intervention and containment measures impacted its dispersal dynamics? And what are the values of key epidemiological parameters? Okay, so I'm interested in developing statistical and computational methods for many different aspects of phylogenetic inference. And to give you an idea of these different aspects, I'm going to talk about some past work that comes from several different research projects. And then at the end, I'll talk a little bit about my research vision and vision for doctoral training in bioinformatics. Okay, so I'm going to talk about coalescent-based models to infer past population dynamics from sequence data and understand how population dynamics relate to ecological and epidemiological factors. I'll also talk about reconstructing the spatiotemporal spread of pathogens, modeling the variability of evolutionary processes, and moving towards real-time evolutionary analysis of epidemics. Okay, and let's start with the coalescent. So the coalescent is a stochastic process that generates a genealogy relating a sample of, let's say, n individuals from a larger population of size n sub e of t. And suppose for a moment that we know the population size n sub e as a function of time. So the coalescent starts at the sampling time and proceeds backwards in time, moving from right to left here, successively merging lineages until all lineages have merged and we reach the most recent common ancestor of the sample, or the root of the genealogy. So the merging of two lineages is called a coalescent event. And at the bottom of this slide, we can see the probability density for the waiting time between coalescent events, conditional on the population size. And by taking the product of such densities, we can obtain the probability density for a genealogy. So the coalescent provides a good approximation of genealogies of samples that arise from an idealized Fisher-Wright reproductive model. And considering this, the population size n sub e might not correspond to the actual census population size, but it's of great interest in molecular epidemiology, for example, as a measure of a population's genetic diversity. And it's referred to as the effective population size. And that's why in the notation we have the subscript E. So the coalescent provides a striking connection between the genealogy and the effective population size, as we can see in this figure. When the effective population size is relatively large, there's a greater waiting time between coalescent events. And when the effective population size is relatively small, there tends to be a shorter waiting time between coalescent events. And this relationship opens the door to inference of the effective population size as a function of time from a genealogy. And that's what we're interested in doing. So coalescent-based models for population dynamics inference are widely used, but they can be hampered by a scarcity of coalescent events for long stretches of a population history. So in a certain sense, the more coalescent events we observe in a given time interval, the more information we have about the population dynamics in that interval. And we see an illustration of this issue in this genealogy on the left-hand side, and on the right-hand side, the corresponding plot of an effective population size trajectory and the uncertainty of the estimate in shaded gray. So the uncertainty, the gray region, increases as we go back in time and encounter fewer coalescent events. So what can we do to mitigate this problem? Well, one idea is to simply increase the sample size, right? If you have a sample size n, you get n minus 1 coalescent events. It turns out that while this helps to some extent, unfortunately, most of the additional coalescent events that you gain from increasing the sample size tend to occur where we already have a relatively large amount of information. What's much more effective, if possible, is to use data that correspond to multiple unlinked genetic loci. So by different genetic loci, I mean, for example, different genes. And what I mean by unlinked here is that the different loci share the same demographic history, but evolve independently and have different lineages. So the idea is, if you double the number of loci, you double the number of genealogies, and then, roughly speaking, you double the number of coalescent events in the time interval in which they are relatively rare. Okay, so this brings us to our statistical model. So we want a flexible, non-parametric approach so that we don't have to make restrictive assumptions about the parametric form, about the shape of the trajectory. So we assume that effective population size is piecewise constant and changes values only at user-specified change points. And then these change points divide up the population history timeline into intervals, so we only have to estimate a vector of effective population size values that correspond to the intervals. So the ingredients of the Bayesian model then are coalescent likelihoods for the observed genealogies. So we're assuming we observe the genealogies for the moment. And then a Gaussian Markov random field prior distribution on the effective population size. So this prior distribution doesn't express any prior beliefs about the overall level of the effective population size, but rather just that its trajectory changes smoothly over time. And the degree of smoothness is determined by a precision parameter, which we'll call tau, and we give this precision parameter a relatively vague gamma prior distribution. Okay, so we want to infer the posterior distribution of the effective population size and the precision parameter conditional on the genealogies. So we can't do this analytically, so we use Markov chain Monte Carlo methods to generate a sample from the posterior distribution, and then we can use this sample to empirically approximate the posterior distribution. So the Metropolis-Hastings algorithm provides a general recipe to simulate a Markov chain, a dependent sequence of random variables that will converge to the posterior distribution. So the way this works is that each iteration of the chain, we use a transition kernel Q to propose new parameter values for the next iteration, and then the new parameter values are either accepted or rejected according to this Metropolis-Hastings ratio. So a lot of standard algorithms update one parameter value at a time, and for a model like this, where there's a large number of correlated parameters, updating one parameter at a time generally leads to slow convergence to the posterior distribution and a very inefficient exploration of the different parts of the posterior distribution. And what this means is that we'd have to run the algorithm for a very long time to generate a sufficient sample. It's much more efficient to update all parameters at once as a block. So what we do is we first come up with a new candidate precision parameter value by rescaling the current value. And then conditional on this precision value, we can generate a candidate log effective population size vector using a Gaussian approximation to the full conditional density. Okay, so far we've assumed that the genealogies are observed. In reality, we don't observe the genealogies, but we can extend this framework to infer population dynamics directly from sequence data. So here we assume that the sequence data for a given locus are generated by a continuous time Markov chain model for a molecular character substitution that acts along a hidden genealogy and depends on a vector of mutation parameters. So these parameters can include, for example, relative exchange rates among characters, across site variation specifications. We'll talk about this in more detail later. So in this model, the coalescent densities act as prior distributions for the genealogies, and they can have an impact on genealogical inferences. For example, the type of coalescent prior you use can impact estimates of divergence times of lineages and the time of the most recent common ancestor of the sample. Okay, so in this framework, we jointly infer the genealogies, the mutation parameters, the effective population size trajectory, and the precision parameter. All of that is jointly inferred from the sequence data. And the nice thing about this joint inference is that we don't condition effective population size estimation on fixed genealogies. We're naturally taking genealogical uncertainty into account. Okay, so to evaluate this model, let's look at some simulations. So we simulate data sets featuring 1, 2, 5, and 10 genetic loci. The true effective population size trajectory, shown as a dotted line here, increases and then decreases. The estimated effective population size trajectories are shown as solid black lines, and the shaded gray regions represent their 95% Bayesian credibility interval regions, the uncertainty of the estimates. And we see that as the number of loci increases, the estimate improves, not only in terms of being less biased and more precise, but also in terms of recovering certain essential aspects of the true trajectory. So first, one aspect, that there's a period of continuous increase followed by a period of continuous decrease. And then a second aspect, where exactly the transition from the increase to the decrease takes place, right? Also, notice that the improvement in inference occurs throughout the population history timeline, but it's most pronounced during the time periods for which we initially, in the one locus case, have relatively imprecise estimates. All right, so a central goal in effective population size inference is to gain insight into the association between the effective population size and potential driving factors, or compare population dynamics with certain ecological or epidemiological indices. For example, information characterizing the spatial and temporal spread of an outbreak. And this has been a major theme in the literature. So to examine the relationship between the effective population size and time-varying covariates, researchers have typically relied on post-hoc analyses. So one common approach has been to simply make informal visual comparisons. More formally, we can employ a generalized linear model framework with the mean of the estimated effective population size trajectory as the response variable. And this is nice because it allows us to estimate an effect size coefficient beta to quantify the relationship and test for statistical significance. But this kind of post hoc approach ignores the uncertainty in the effective population size estimate. And it also neglects the opportunity to use covariates when appropriate to inform the effective population size estimate. Okay, so why is ignoring the uncertainty in the effective population size estimate a problem here? So for one, it can lead to effect size coefficient estimates that have inflated precision. On the other hand, it can erroneously rule out significant associations by overemphasizing the incompatibility between the covariate and effective population size trajectories during certain time periods. So here, for example, we have the mean effective population size trajectory in black and covariate in blue. And if you compare the two curves, we see a strong correspondence for much of the timeline, but there's a great divergence between the two on the left side of the plot, and then also a notable divergence on the right side. Okay, but when we consider the uncertainty of the effective population size estimate, shown in shaded gray, we see that during the time periods where the mean effective population size and covariate exhibit divergent behavior, there's a great deal of uncertainty about the effective population size. So one might argue that the strong correspondence that we see when we have a lot of information about the effective population size should, in some sense, mean more than the discordance that we see when there isn't much information about the population dynamics. So by incorporating covariates directly into our model, we can simultaneously infer the effective population size and covariate effect size coefficients, while naturally accounting for the uncertainty of the effective population size. If there is a significant association, this also allows the covariates to inform the effective population size estimate. And this can be especially valuable for certain time intervals where there isn't much signal in the sequence data alone. Okay, so we want to model the effective population size for a given time interval as a log linear function of covariates measured during that interval. And we want to model the intercept term as a Gaussian Markov random field to impose temporal dependence at neighboring intervals. And we can get what we want here by assigning the log effective population size vector a Gaussian Markov random field prior distribution with the inner product of the covariates and effect size coefficients as a mean. Okay, so we can use this framework to examine the dynamics of the West Nile virus in North America. So here we consider a data set of 801 genomic sequences sampled between 1999 and 2016. And as a covariate, we consider the temperature of the affected area. So we infer a significant positive association between the effective population size and temperature. And this is consistent with what we know about the virus and its hosts. For example, mosquitoes are one of the main hosts for the West Nile virus, and higher temperatures have been shown to directly impact the mosquito life cycle. For example, by accelerating larval development, decreasing the interval between blood meals, and prolonging the length of the mosquito breeding season. So in the plot here, the covariate values are shown in orange. The solid black curve is the effective population size estimated only from the sequence data without any covariate, and its 95% Bayesian credibility interval region is represented by shaded gray. The solid blue curve is the effective population size inferred from both the sequence data and the covariate data, and its corresponding Bayesian credibility interval region is in shaded blue. So what we see is that including the covariate in the model impacts the estimate of the effective population size to more closely reflect covariate trends and be more precise while still being compatible with what we infer strictly from the sequence data. And what I mean by that is that the blue region inferred using covariate data as well as sequence data is almost entirely contained within the gray Bayesian credibility interval region, which was inferred only from the sequence data. Okay, so now we want to turn to models for the evolution of continuously varying traits on phylogenetic trees. So these traits could be phenotypic traits, which are of interest in phylogenetic comparative methods, which focus on relating observed phenotype information to an evolutionary history. And a proper understanding of patterns of correlation between traits can be achieved only by accounting for their shared evolutionary history. These traits, so to speak, could also in fact be geographic locations associated with sampled sequences, which would then allow us to reconstruct the spatial and temporal spread of a population and connect it to its evolutionary history. And these kind of phylogeographic techniques have allowed researchers to better understand the origin and spread of infectious diseases. Okay, so suppose we observe n continuous trait values, each of dimension k. We can think of these observed values as corresponding to the tips of an unknown but estimable phylogenetic tree. And we also assume that we have unobserved trait values at the internal nodes of the tree. So we can think of the observed trait values as being produced by a Brownian diffusion process that starts at the root of the tree and then proceeds down the branches. So here, the displacement of a trait value along a branch. So what I mean by that is the trait value at a child node minus the trait value at the parent node, which connect the branch. The displacement of a trait value along a branch is normally distributed with a mean of 0 and a variance that's proportional to the branch length. And this kind of standard correlated Brownian diffusion is maybe the most widely used model for continuous trait evolution, but it's also very restrictive and it might not adequately characterize the true process. One of the major restrictions is that these displacements are assumed to have a mean of 0. What we'd like to do is relax this restriction by introducing an estimable mean term that can account for and quantify directional trends. So in particular, we want the displacement mean to be equal to a branch-specific trend vector called mu I multiplied by the branch length, ti. Unfortunately, allowing each branch to assume its own unique trend vector can lead to problems where different collections of trends yield the same likelihood. So to ensure model identifiability while still permitting variation in trends, we introduce some restrictions that give us what we'll call the relaxed directional random walk. So the restriction is that whenever a branch splits into two child branches, either both child branches inherit the trend associated with the parent branch, or one of the child branches assumes a new trend while the other child branch inherits the trend of the parent branch. Okay, so what we would like is for the data to determine the type of trend transfer that is appropriate at each node. And we can accomplish this by adapting ideas from Bayesian stochastic search variable selection to efficiently explore the different possible trend configurations corresponding to an evolutionary scenario. So here we can let gamma I denote a branch-specific trend change indicator. So it's equal to 1 if a trend change occurs for that particular branch, and equal to 0 if no change occurs. The trend for a given branch then is equal to the trend assumed by the parent branch plus a trend difference, alpha I. The trend differences, alpha I, are assumed to be independent and normally distributed with a mean of 0 and a variance that's proportional to the trend change indicator, gamma I. So if there's no trend change, meaning that gamma I is equal to 0, the prior variance shrinks to 0, and this forces the trend difference alpha I to be equal to 0 in the posterior distribution. And we can explore this joint space of trend change indicators and trend differences through Markov chain Monte Carlo. One more important computational detail is evaluating the likelihood of the observed trait values. So it's possible to show that the likelihood has a multivariate normal density with a covariance matrix of dimension n times k, where n is the number of observed trait values and k is the dimension of the trait. And this suggests a computational order of n cubed times k cubed to evaluate the density. So notice here that n is typically going to be a number in the hundreds or thousands, whereas k will typically be a number in the single digits. It turns out that it's much more efficient to evaluate this, this likelihood using a dynamic programming approach. So we start with the joint density of all trait values, observed as well as unobserved, and then we recover the density of the observed values by integrating out the internal node values one at a time in a post-ordered tree traversal. So this dynamic programming algorithm reduces the computational order from n cubed times k cubed to n times k squared. And because we have to repeatedly evaluate the likelihood in MCMC algorithms for posterior simulation, this can make a really big difference in allowing the model to scale efficiently to data sets that feature thousands of observed trait values, as is often the case in virus evolution. Okay, so let's look at an example. Faria et al. Explored the early spatial expansion and epidemic dynamics of HIV-1 in Central Africa by analyzing sequence data sampled from countries in the Congo River Basin. And they used a discrete phylogeographic inference framework on 466 HIV-1 sequences sampled between 1985 and 2004. And they showed, among many other things, that the pandemic originated in Kinshasa in the 1920s and then spread to other population centers in sub-Saharan Africa. So we want to analyze this data set with several different Brownian diffusion models for continuous phylogeography and compare the results. So notice here that the continuously varying traits in this instance consist of bivariate latitude and longitude coordinates. Okay, and here we have a map with the sampling locations and the number of sequences sampled in each location. So given an origin of Kinshasa, we might expect general directional trends to the south and east. If we allow for trend changes, we might expect some trends along the phylogeny to the north to Bomanda and Kisangani, and perhaps also to the west to Point Nord. All right, so here we have the results for 3 different models: Brownian diffusion with no trend parameter, Brownian diffusion with a trend that's restricted to be constant along the phylogeny, and Brownian diffusion with a relaxed trend parameterization. So we infer significant trends to the east and south, and under the relaxed trend model, we infer a mean of 28.13 latitudinal trend changes and 1.48 longitudinal trend changes. Okay, so let's consider the displacement variance rates, which correspond to sigma 11 and sigma 22 in the displacement distribution in the center of the slide. So if you look at the second row of the table, notice how much the displacement variance rate for the longitude is reduced, going from a model with no trend to one with a constant trend. And if you look at the fourth row of the table, notice how much displacement variance rate for the latitude is reduced when we go from a model with a constant trend to one with a relaxed trend. So these results show how failing to adequately model trends can result in the underlying trend being conflated with the displacement variance rate and also inflating the displacement variance rate. On the other hand, flexible modeling of trends can lead to a more precise, detailed reconstruction of dispersal dynamics. Okay, now I want to discuss a flexible class of models for sequence evolution. So as we touched on before, sequence evolution is typically modeled by continuous time Markov chain models of character substitution that act along the branches of a phylogenetic tree. And the Markov chain is characterized by an instantaneous rate matrix that specifies the relative character exchange rates. So early phylogenetic reconstruction methods assumed a single substitution model that acted independently and identically across the different sites of a multiple sequence alignment and the different lineages of a phylogenetic tree. However, the evolutionary processes that act at the molecular level are highly variable. For example, it's well established that the evolutionary process can vary among the sites of a multiple sequence alignment, and it's become common to model among site variation in the relative substitution rate as well as in character exchange rates. However, among site heterogeneity might also be time dependent and lineage dependent. Some sites evolve quickly in some lineages and slower in other lineages, while different patterns are observed at other sites. So, for example, under the covariant hypothesis of Fitch and Markowitz, at any given time, some sites might be invariable due to functional or structural constraints, but as mutations are fixed elsewhere in the sequence, these constraints might change, and sites that were previously invariable may become variable, and vice versa. So our goal is a general framework that combines site-specific and branch-specific variability. So suppose we have k evolutionary models, where each evolutionary model is determined by a substitution model characterized by an instantaneous rate matrix, Qi, and possibly a relative substitution rate multiplier, rho I. The evolutionary process for each site can switch between these k different evolutionary models along the phylogenetic tree. And the switching is determined by an auxiliary continuous time Markov chain. And this results in a Markov modulated Markov model that can also, in fact, be described as a continuous time Markov chain. And we can construct its instantaneous rate matrix from the evolutionary model parameters and the switching model parameters. Okay, so let's look at an example of where this type of Markov modulated approach can make a big difference. So nucleotide composition can vary greatly within and among organisms, and as a result, unrelated taxa sharing similar base composition can sometimes be grouped together within a clade. So here we consider the 16S ribosomal RNA of 5 bacterial sequences, and the true tree topology of these sequences is believed by experts to group Deinococcus radiodurans and Thermus thermophilus together to the exclusion of the other 3 sequences. However, it's known that most standard phylogenetic inference methods erroneously group Deinococcus radiodurans and Bacillus subtilis together due to their similar base composition, in particular, a low GC content. So here on the left, we see a tree estimated using 2 standard nucleotide substitution models, the HKY model and the GTR model. So as you can see, the Deinococcus radiodurans and Bacillus subtilis sequences are erroneously clustered together with posterior probability 0.649 for HKY and 0.863 for GTR. On the right, we see an estimate from a Markov modulated Markov model comprising 2 HKY substitution models. And it yields the correct clustering of Deinococcus radiodurans and Thermus thermophilus sequences with a very high posterior probability of 0.959. All right, so now I want to talk about online inference and the scenario that motivates it. And what I mean here by online inference is an inference framework that can, in some sense, accommodate data that arrive bit by bit in a serial fashion, rather than requiring that all of the data are available at the beginning of the analysis. So advances in sequencing technology have enabled genomic surveillance during outbreaks in close to real time. And this type of continuous stream of new sequence data creates the potential for phylogenetic inference to assume a more prominent role in the public health response by providing timely, updated inferences during the course of an ongoing outbreak. However, the current state of Bayesian phylogenetics is such that we assume that the data set is complete when we start the analysis. If new data become available after starting the analysis, we have to stop the analysis, discard our results, and then restart the analysis from scratch on the expanded data set. And given the long computational runtimes frequently encountered in Bayesian phylogenetics, this is a major obstacle. So to remedy this, we propose an efficient online approach that updates previous inferences as new sequence data become available. All right, so one of the major hurdles in using Markov chain Monte Carlo methods is what's called the burn-in. So the burn-in is a transient phase at the beginning of the simulated Markov chain before it converges to the stationary distribution, during which simulated values retain the influence of the starting values of the chain. And the burn-in is typically thrown away to avoid biasing inferences. The length of the burn-in can depend upon how far away, in some sense, the starting values are from sort of the heart of the true posterior distribution. And for phylogenetic models on realistic data, the burn-in can be extremely long and represent a large proportion of the iterations that are necessary to generate a sufficient posterior sample. So minimizing the burn-in could substantially reduce the computation time And online inference represents a unique setting where this just might be possible. So our strategy is that whenever new sequences become available, we want to use inferences up to that point, along with the newly arrived sequences, to construct what we hope will be good starting values for an analysis of the expanded and large data set. And what I mean by good starting values here is starting values that are either in or close to a high probability region of the target posterior distribution. So here's how our online inference setup works. So after a chain has converged and after the arrival of new sequences, we interrupt the analysis if it's still running. Next step is to extract the parameter values from the last iteration in the interrupted analysis. Then we insert each of the new sequences into the tree that we just extracted from the interrupted analysis. For each new sequence, we compute its genetic distance from the sequences that are already in the tree, and the minimum genetic distance determines the height at which to insert a new common ancestor node for the new sequence and its most genetically similar sequence. The next step is to impute plausible values for new parameters that are introduced to accommodate the new tree branches. For example, relaxed clock model rates. For parts of the model that don't assume increased dimensionality, the old parameter values are simply carried over. All right, so now we've finished constructing what we hope is a good starting parameter value, and the last step is to resume by starting an analysis of the expanded data set from this good starting value. And to see how much of a difference this can make, we consider a data set of 1,610 whole genome sequences from the West African Ebola virus epidemic of 2013 and 2016. And we compute updated inferences at different time points of the epidemic using two different approaches. First, a standard approach where we just analyze the complete data set from scratch at each time point. Second, an online inference approach where we use the results from the analysis of all of the data that were available by the previous week, along with newly arrived data from the current week to construct starting values. All right, so here we have the data flow in terms of total sequences available during each week, recreated in the background of the plot in gray bars. Dark gray bars show the data corresponding to the 5 time points at which we compute updated inferences. So the plots show the average burn-in over 5 replicates required by standard analyses, represented by circles, and online analyses, represented by diamonds. So notice that all the diamonds are close to 0. Solid lines correspond to burn-in estimates based on visual analyses of trace plots, while dotted lines correspond to burn-in estimates based on maximizing effective sample size values. And we see that under the online approach, the burn-in is, in all the cases we consider, virtually eliminated. So to put the reduction in burn-in into perspective, we can translate it to how much computational time we save. So notice here that the y-axis is on a log scale. So the time saved varies by replicate and also by the computational resources used. So under a standard CPU, the average savings for the latest time point are about 590 hours, which is more than 24 days. For the 2 time points before that, the average savings are more modest, but still substantial, 90 hours and 25 hours. If you have a state-of-the-art GPU, you won't save as much time, but the savings are still very substantial. For example, for the latest time point, we save on average about 120 hours or 5 days. Okay, so all of the work that I've discussed here has been implemented in BEAST, an open source software program for Bayesian phylogenetic inference. So finally, I've also been fortunate to work with a number of great collaborators who I'd like to acknowledge. And this work would not have been possible without their help. And I want to just talk briefly about my vision for research moving forward. So the motivation for my work is to substantially enhance the capabilities and impact of phylogenetic inference in infectious disease epidemiology. But I want to add that the statistical and computational methods I plan to develop will be much more widely applicable to study a broad range of questions in evolutionary biology. And my strategy for achieving my goals has several parts. So first, to strive for more accurate phylogenetic inference through modeling that's more flexible and or in some sense more biologically realistic. Second, to develop new frameworks that integrate epidemiological and ecological information in addition to molecular sequence data and can help us better understand the interplay of evolutionary, epidemiological, and ecological processes. Third, to accompany these new statistical methods with efficient algorithms that allow them to scale to large data sets. And finally, for maximal impact, to provide accessible software implementations with tools for analysis and visualization of results. And as far as doctoral training, this research has several different and important aspects, new statistical methods, computing and software development, and important questions in biology that motivate everything. And I'm excited to work with students who come from different backgrounds and are interested in different aspects of this program. Okay, thank you very much. I'm happy to take any questions.
Thank you, Dr. Gill. It looks like Nathan, you have a question. Do you want to unmute yourself?
Yeah, sure. That was a really cool talk. I just have two questions, if that's okay. My first question is about the online method, or like you're taking in on the fly data and creating trees from that. So you said you were using like an assumption of like interval time, and my question is, if you had like data that was like every other week and then you swapped it to like a monthly scale, would that affect like the variability of it or the model, or like would it affect the accuracy of the model by just changing up like the interval, or does it require like an exact interval? If that's kind of my question.
I don't think that the interval matters so much as how many new sequences you're trying to add. So, for example, if you have a certain number of sequences that you've already analyzed or that you're in the process of analyzing, right? And then there's some new data that become available, and you want to accommodate the new sequences into your analysis. So, if you're analyzing like 100 sequences and then you suddenly want to add 100 more, it's not going to reduce the burn-in to the same extent as if you're analyzing 100 sequences and you only want to add 5 more or 10 more or something. So it really just depends upon the amount of data that you want to accommodate into your analysis and kind of the best way to add that. Does that make sense?
No, that makes a lot of sense. Thank you so much. And I'll let the other people ask questions, and then I will, if we have time, I'll ask you the second one.
Okay.
Dr. Summers?
Hi, that was way over my head, but very interesting, and it stimulates a couple of questions I have from a very different zone of infectious disease, and that's mobile genetic elements. And is it the case that these tools that you've devised would be equally applicable to classical the gradualist point mutations, single nucleotide polymorphisms, and homologous recombination, and also to very non-homologous recombination and random occurrence of recombination that's characteristic of the mobile genetic elements that are major players in the spread and occurrence of antibiotic multi-resistance?
Yeah, thank you. So I think that so these methods are applicable to any sort of measurably evolving population. If that makes sense. Not just rapidly evolving pathogens, but any sort of population where we have sequence data, molecular sequence samples, from different points in time that allow us to sort of reconstruct evolution in some sense.
So that's whether or not the variation is occurring as point mutations, or slabs of entire genetic material being exchanged. Like whole genes at a time being mobile on their own.
Yeah, to be honest, I'm not sure I properly understand. It might be, I think it might be a bit over my head what is meant by that.
Well, I would like to consider that problem with you, so I will get in touch.
Okay, yeah, thank you. I'm sorry I can't answer that better
Well,
right now.
that's okay. Thank you.
Dr. Mrazek?
Hi Mandev, I'm sorry if you hear my air conditioning noise here. I was wondering, when you're talking about population and population size, what exactly that means? Because I think at the beginning you have the last common ancestor, which presumably is a single population, but as it starts diverging and branching into those variants or strains or species or whatever you mean to call it, is it reasonable to assume that it is still a single population? Or do they split in distinct population? And if they do separate into distinct populations, how would that affect your model?
Thank you. That's a great question. So this version of the coalescent that I base my development on, it's a simplified version that assumes that you don't have subpopulations with perhaps migration between these different subpopulations. So more other coalescent-based models that account for that have been developed, and so other people have worked on extending those models to integrate into a phylogenetic inference framework. And I think those are some interesting things that I would like to look at, but the work that I discussed here doesn't account for that. So that is a limitation.
Okay, thank you.
I have a quick question. How do you make, in what cases would you rather use a discrete phylogeographic model rather than a continuous phylogeographic model, and vice versa? Like when would each one be more useful than the other?
Thanks. So
Like, what
it
types
really
of
depends
questions?
on the nature of the data that you're working with. So sometimes the samples that you have, if you look at the sampling locations, are more continuously distributed, or so in that case, a continuous phylogeographic model might be more useful. Also, in situations where the spread is kind of in a region where that makes sense. Like, for example, you wouldn't want to use continuous phylogeography where you have samples taken from very disparate regions which are separated by oceans, for example, right? In those types of situations where the sampling locations are much more discretized, where it doesn't make sense to have like a continuous reconstruction that goes over the Atlantic Ocean or something like that, then a discrete phylogeographic inference framework is more appropriate. It also can depend upon this scientific question you're trying to answer. Like the model parameters that you will infer using these different approaches will be different. In a discrete phylogeographic inference framework, you'll infer these transition rates from one location to another, so you can kind of focus in on that. Whereas if you have this continuous phylogeographic framework, you're just going to be inferring more general diffusion rates. So yeah, it depends on a lot of things.
Thank you. And we are coming up on time, but Nathan, if you want to ask the last question.
Yeah, this is also a question about like the runtime of the fusion part of your project. I would just ask, I just want to know, like, when it comes to like getting that return on runtime and making it really efficient, does it require like the phylogenetic tree to be balanced at all? Is it robust to like one branch being like really long and the other one being short, or can you still like get that return on runtime?
So are you talking about the online inference framework?
The fusion the part where like you were saying like it was like n cubed to k cubed to like n to k squared or something.
Oh, right. Okay, yeah, I see what you mean. Yeah, I mean, I don't see where that would depend upon specifically on that balance that you described. I don't think that would make a difference. Am I missing something here? Is there a reason you think that would?
The only reason I'd say that is like maybe you're utilizing like a heap shortcut of like depth first search, but like it's been a while since I did like tree traversal and like stuff, so I could be mixing up some stuff. My bad.
Yeah, I don't think so. I mean, I could be wrong, but thinking about it right now, I don't think that would impact things.
All right, thank you so much.
Well, thank you again, Dr. Gill, for your presentation. If everybody wants to just look a little thank you clap. But that'll conclude the seminar for today.
Okay, thank you all for attending.